So it gives me great pleasure to introduce Kimberley Kendall, who's an academic and also a training in general adult psychiatry from South Wales. She studied genetics, medical genetics and medicine at Cardiff University, and then completed a PhD locally at the neurodevelopmental on a subject, which is just because you want to talk about neurodevelopmental copy number variants at the MRC Centre for Neuropsychiatric Genetics and Genomics in Cardiff. And she's currently works in a rehab psychiatry and assertive outreach, and also working mostly in individuals with psychotic disorders. And her main interests are in finding out the impact of psychiatric genetics and how they can improve patient care. So over to you, Kimberley, and thank you for coming. I'm looking forward to your talk.
Thank you very much, and thank you very much for the invite to speak. As you just mentioned, obviously, my name is Kim, and I'm a psychiatrist. I'm actually on the WCAT scheme, which is a combined clinical academic scheme, and I've just come to the end of my ST4 job in rehab. I'm a postdoc then in psychosis and psychiatric genetics in particular, and my favourite topic in the world is copy number variants, which perhaps makes me a little bit sad, but never mind. Okay, so what I'm going to talk to you about today is kind of an extension of what I did my PhD on. So you may have seen publicised that the talk was called The Phenotypic Spectrum of Neuropsychiatric CNVs. This is essentially what the talk is, but I've added some extra bits on how we might be able to use this work to benefit patients a bit more, to try and make it more translational. So this is the plan of what I'm going to cover. I'm going to start with a little light revision. Now, I make no apologies whatsoever for this, because I give this talk really quite frequently, or this kind of talk. And the audience is often very different in terms of how long it's been since they did any genetics, and certainly their level of comfort with genetics. So I don't think it causes any problems at all to make sure we're singing from the same hymn sheet. And I certainly don't mean to patronise, so apologies if you find the beginning bit a little basic. I'll then move on to copy number variants and what we know really about their roles in psychiatric disorders. Then we'll move on to a little bit about non-psychiatric phenotypes, because they do have implications for our patients. And then finally, we'll end on some translation, including some ethical issues. So let's start with a little revision. So as you likely know, DNA is a double-stranded molecule consisting of a phosphate backbone and complementary bases. So cytosine pairs with guanine, adenine with thymine. And DNA is vital really for the production of proteins from which our bodies are made and function. Genes are sections of DNA which encode proteins. So they go on to make proteins. Exons are the parts of the gene which will be transcribed into messenger RNA for later translation into a protein. The introns are the bits between the exons, which we thought were junk originally, but it turns out they're not. They're involved in processes such as gene expression. And the promoter, which is at it's to the left of your screen, but we call it the 5' end of the gene, and that's involved in controlling transcription. And DNA is housed with associated proteins in chromosomes, and we all have 23 pairs. Well, vast majority of people have 23 pairs. So we have 22 pairs of what we call autosomes. So that is basically non-sex chromosomes, and 1 pair of sex chromosomes. So XX for females, XY for males. So this karyotype, so this whole thing is called a karyotype. So this is from a male, because you can see X and Y, and you'll notice that the Y chromosome is quite dinky compared to the X. It contains far fewer genes. Okay, so psychiatric disorders. So you'll hear the term genetic architecture used a fair amount by people like me, to be honest, and sometimes we forget to explain what we mean by it. It's basically the structure of the genetic basis of a disorder. So you'll likely be really familiar with Mendelian inheritance. So, you know, the autosomal or dominant, X-linked, recessive, all of those kind of terms. None of those apply to psychiatric genetics. Kind of unfortunately, actually, because it just makes things an awful lot more complicated. So the genetic architecture of psychiatric disorders is complex. You don't actually ever inherit a psychiatric disorder. You inherit an increased predisposition to develop that disorder. And that risk is comprised of common variation, which is of individually small effect, and rare variation with far greater effects. And it's CNVs which belong to this latter group, so rare variants with large effects. So let's move on to copy number variants, or CNVs. CNVs are any region of the genome which occur at variable copy number in comparison with a reference sequence. So that's quite a convoluted way of saying anything where there's a section where there's too many copies or there's fewer than you'd expect. And copy number variants can get quite complicated because chromosomes can swap bits of genetic material quite commonly. But really, what I'm referring to when I say CNVs in the context of this presentation is the deletion or the duplication of more than 1,000 base pairs of DNA. So if you think about it, that's an awful lot of genetic material to lose or gain. And this fits with their effect sizes. Now, obviously, effect sizes depend on what you're looking at and what you're considering. And the effect sizes of CNVs quite clearly will depend on where they occur in the genome. But the size of CNVs, the fact they're so large, means that they have a far greater chance of hitting a potentially important gene than, say, a single base pair change. Just to explain, penetrance, which is this term here, is the term used to describe how much a variant is expressed in an individual. And penetrance depends really on what you think, what condition you're talking about, because CNV is predisposed to an awful lot of conditions. So let's take this as an example, the DiGeorge VCFS, or velocardiofacial syndrome locus on the chromosome 22. This is one of the more highly penetrant CNVs. So all of these are actually schizophrenia CNVs, but this one in particular is highly penetrant. You'll see that it really quite impressively increases the risk of schizophrenia compared to your background population risk of about 1%. But actually, you know, there are a fair number of people who don't get schizophrenia, but they have a massively increased risk of developmental delay, ASD, congenital malformations. And this is true of basically all of the neurodevelopmental CNVs. Excuse me. And that quite neatly brings us to the concept of the neurodevelopmental spectrum. So you'll probably be aware of the hypothesis of the neurodevelopmental spectrum in the context of childhood onset disorders like intellectual disability and autism. I think that was probably around from around about the 80s. But it really was in about the 2000s that researchers and psychiatrists started to suggest, well, should this be expanded to include schizophrenia? In this model, the greatest level of neurodevelopmental impairment is observed over here on the left with intellectual disability. And as you move through ASD and schizophrenia, that level of neurodevelopmental loading decreases until we reach major depressive disorder, which perhaps we don't really think of as a neurodevelopmental disorder at all. Other features of these disorders also appear to vary in a spectrum-based manner. So the greater the level of neurodevelopmental loading, which is towards this end, the greater degree of cognitive impairment, the earlier age of onset, the greater functional impairment, and the greater role for your rare, quite damaging mutations, such as copy number variants. And so the people who developed this model, multiple researchers who were involved, also suggested that some of these other symptoms, such as negative symptoms and positive symptoms, may vary in a similar manner, although obviously in a different pattern to the cognitive impairment. So let's move on to what we know about copy number variants in psychiatric disorders. So CNVs are established risk factors for disorders on the neurodevelopmental spectrum. That much is fairly certain, to be honest. But they've all, obviously, different studies have adopted slightly different approaches, so have implicated different types of CNVs in risk of their disorders. So within intellectual disability, there have been increased rates of rare CNVs reported, quite large in some of the studies. So you're talking over 500,000 base pairs. And that is more for deletions than duplications. So we think, and well, we know based on the evidence, that deletions are more deleterious than duplications, which perhaps isn't that massively surprising. In fact, it's probably quite intuitive. Intellectual disability CNVs often occur at 55 loci or genomic locations associated with known genomic disorder regions. So these are the regions that would pop up in a medical genetics clinic, and they're often recurrent. So that means that they are flanked by low copy repeats, so runs of repeated sequences, which can cause slipping when you get DNA replication, and that just forces errors. Increased rates of small CNVs have been reported in ASD, although this does seem to have been driven by people with ASD and co-occurring intellectual disability. But we do know that there are several individual CNV loci which have been reported as being enriched for de novo CNVs. Now, I'll explain some of these terms as we go, because just in case we have some students with us, de novo CNVs are those that have occurred in a particular family in one individual for that first time. So the CNV hasn't been inherited from their parents. And this has been reported at 6 loci for ASD, and you'll probably recognize that some of them appeared on that schizophrenia chart a little while ago. And that's something that's quite stark from all of the neurodevelopmental CNVs. They're decidedly non-specific. One more small note on nomenclature, for those who perhaps haven't got much of a background in genetics. The number before the letter is the chromosome number. The letters Q and P are the arm of the chromosome, and the easiest way to remember that is P is for petite, so it's for the short arm, and then this number afterwards is the band of the chromosome arm. So it just helps you to kind of picture where these things are. Increased rates have been reported in ADHD as well. There's less literature on ADHD, but an excess has been reported at 2 different sizes and at 2 individual loci. Interestingly, these were both duplications. I don't think they've actually been replicated, but that might be due to sample size and the amount of work that's done in a particular disorder. Schizophrenia. Now, I mentioned the expansion of the neurodevelopmental spectrum to include schizophrenia, and there is a lot of evidence for the role of CNVs in this disorder. So broadly, an excess of the CNVs has been reported as deletions and duplications at over 100 kb, but there's now robust evidence for about 12 loci to be individually associated with schizophrenia. And the first of these was that CNV that I mentioned a while back, which was the 22q11.2 deletion syndrome, CNV. And this was really implicated in schizophrenia. I think it might have been in the 90s, when researchers and psychiatrists noticed that individuals with this particular syndrome, because it was identified as a syndrome, had quite starkly raised rates of schizophrenia. And then they carried out more systematic searches and found that actually they do have around about sort of 25% chance risk of the disorder compared to a background risk of 1%. And the second was exonic deletions in the neurexin gene, which is on chromosome 2. And since these, a further 5 deletion CNVs have been associated with schizophrenia and 5 duplications. And these CNVs have all been selected on the basis of being associated with schizophrenia at a genome-wide level of significance. So we're talking really quite robust findings, because when you test CNVs genome-wide, you carry out so many tests that you have to do lots of correction, and these are all corrected and still significantly associated. And I mentioned earlier on that these CNVs have large effect sizes. The odds ratios for all of these CNVs range from around 2 to in the 50s. So these are all neurodevelopmental disorders, I would argue. What about those that we don't actually normally consider to be neurodevelopmental in origin? So the evidence for bipolar affective disorder is, well, there's less of it. It's less clear and it can be conflicting. This seems to be largely due to sample size and because of the considerable clinical heterogeneity of the disorder. I mean, that's fairly intuitive, isn't it? That you've got bipolar 1, bipolar 2. Often included in these studies is the bipolar variant of schizoaffective disorder. And this particular study done by Alexander Charney et al. Really lended weight to this. So they analysed bipolar spectrum disorders, so those 3 that I mentioned previously. And when they treated them as one group, there wasn't any evidence for an increased rate of CNVs. But then when you started segmenting it up into bipolar 1, 2, and schizoaffective bipolar, there was an increased rate of CNVs with the bipolar in the bipolar subtype of schizoaffective disorder when you compare it to bipolar 1. So I mean, that study doesn't provide conclusive evidence, but I think it's interesting that the CNVs were implicated in the disorder which is phenotypically closer to schizophrenia. So let's move on to major depressive disorder, which I don't think anyone would consider to be a neurodevelopmental disorder. Between 2010 and 2019, multiple depression studies were carried out. They were a little bit underwhelming in the sense that they produced conflicting evidence. A lot of results didn't replicate, and it was all slightly messy by the end of the 2010s. And then that again, like bipolar, was probably due to the fact that the studies were underpowered and the disorder is really heterogeneous in terms of its phenotype. In fact, let's face it, it's probably more than one disorder. So in 2019, 2 studies came along which bucked this trend somewhat. So this first study was carried out by Zhang et al. And they carried out a meta-analysis of about 5,500 cases and 6,500 controls. And they found that there was an increased burden of short deletions in major depressive disorder. In the same year, I led a study, which was one of the chapters of my thesis, looking at the role of neurodevelopmental CNVs in the risk of depression. So so you remember I mentioned 55 genomic regions for intellectual disability. I took a list of 53 CNVs from these regions. The reason it's 53 rather than 55 is that some of them were too common to be included. So these are all rare individually, and they occur at a frequency of less than 1%. So I took the 53 associated with intellectual disability, developmental delay, and autism, and looked at them in the context of depression risk in individuals in UK Biobank who haven't developed a neurodevelopmental disorder. And that's the important bit. So I found that these CNVs were associated with the risk of self-reported depression. This is a self-report of a doctor-made diagnosis. And the odds ratio was about 1.3. And I repeated it in 2 more conservative variables. So where I constructed my own from self-reported depression and antidepressant prescription. And I used ICD-10 codes from hospital admissions. And I found that when I repeated the analysis in these additional variables, the odds ratio actually increased, although the confidence intervals overlap, so you can't make too much of that. But the important thing is that the result was still there. Now, these results raised questions when I first presented them. We know that these CNVs are associated with disorders characterized by neurodevelopmental impairment, but all of these individuals, by definition, because that's how I carried out the analysis, didn't have neurodevelopmental disorders. So you couldn't engage in diagnostic overshadowing and blame a neurodevelopmental disorder. So why would they have an increased risk of depression? And there were a couple of things that we thought of along the way. Some work I'll present in a second on cognition shows that individuals who carry these CNVs have impaired cognition relative to non-carriers. So is it that a kind of subclinical cognitive impairment might impair your ability to live your life and cope with stressors? Perhaps that would increase your risk of depression. Or would you engage in more difficult lifestyle behaviors, for instance? So, or is it just that CNVs increase the risk of depression? And there's a direct link. So I carried out some structural equation modeling analyses where I tried to find variables that could explain the association, and I looked to see how much they did. CNV carriers had poorer physical health, higher rates of smoking and alcohol consumption, lower educational attainment, and higher rates of social deprivation than their CNV non-carrying counterparts. However, when you include all of these together, it only accounts for 20% of this relationship between neurodevelopmental CNVs and depression. So either we're looking at a considerable direct link between the two, or there may well have been other variables that we didn't have, that could potentially explain the association, or perhaps it's both. Now, the beauty and the wonder of the UK Biobank sample is its size. And so for the first time, we had really good power to look at quite a few of the individual CNV loci for association with depression risk. And these are the individual CNVs with more than 5 observations. And if you look at the ones with the little letter B, so, oh, sorry, not that one. So I found robust associations which survive correction for multiple testing for associations between 3 CNV loci and depression risk. The first was deletions on chromosome 3 at 3q29. The second was duplications on chromosome 8. And the third was the duplication version of the DeGeorge syndrome CNV. So that's the 22q11.2 duplication. So we've spent some time discussing links between rare CNVs and disorders themselves. What if a patient already has a psychiatric disorder? Is there much point in finding out their CNV carrier status? Will it make any difference to them at all? And the answer to this question will likely vary for individuals, to be honest, but it would be helpful to understand a bit about the associated non-psychiatric phenotypes, of which there are several. So let's start with cognition, which I referred briefly to just now. So as we know, large rare CNVs are associated with disorders characterized by neurodevelopmental impairment, which include cognitive impairment. But you've likely worked out during this presentation, because I've referred to it, that not everyone who has a CNV develops a neurodevelopmental disorder. So are we talking about completely unaffected individuals? Are we saying that the phenotypic spectrum can include no effect at all? Or are these people more subtly affected, which can help us understand a bit more about how these CNVs might work? And this again is a question we asked of the UK Biobank. So I examined the results of 7 cognitive tasks for individuals with these neurodevelopmental CNVs, but no neurodevelopmental disorders. Now, for various reasons, I can explain, we split the 53 CNVs into the 12 schizophrenia CNVs in green, and the rest were red, so they're neurodevelopmental CNVs. And the reason for that was that we were trying to replicate some previous work, but essentially you could just combine them and it would be the same thing. And for reference, we included people with schizophrenia from UK Biobank. Let me orientate you to this graph. So the 0 line, so I've standardized all of these results according to non-carriers. So your 0 line is your result for people who don't carry CNVs. So anything below the line is a deficit, so they're performing more poorly. So you can see across almost every task, people who carry a schizophrenia or an other neurodevelopmental CNV perform more poorly than CNV non-carriers. However, they don't perform quite as poorly as those with schizophrenia. So they have that kind of intermediate stage of performance. And this pattern extended to qualifications. So again, just to orientate you to the chart, carriers of the CNVs, the two groups that I previously mentioned, are in black and grey. And then the CNV non-carriers were in white. So individuals who carried CNVs were far less likely to end up in the higher qualification category of college and degree. And I'll go back to this graph in a second. And it was the same pattern for jobs. So this major job group is defined by the Office of National Statistics, and group 1 is the highest. So you're talking company directors and things like that for number 1. And group 9 is your elementary jobs, things like that. I would actually say what is more useful from this is the fact that so many of them did achieve so highly. So it's all very well saying that carriers of CNVs are less likely to get a degree, but in the UK Biobank, almost 25% of them did. And I think that's really important because we previously saw these CNVs and thought, oh, they're really nasty CNVs, they're syndromic, they cause really bad impairment. Not for these people. Although still, if they didn't have the CNV, then perhaps they wouldn't be quite so impaired. So I just think that's an interesting thing to point out. And it's important for genetic counselling. So this work replicated and extended some work from an Icelandic group who originally showed some of the cognitive stuff, and some work on educational attainment done in the Estonian population. So all of my work done so far was done in individuals without neurodevelopmental or psychiatric disorders. But what about people with things like schizophrenia? Excuse me. A colleague of mine, Dr. Leon Hubbard, carried out a study of people with schizophrenia with and without CNVs, and found that of people with schizophrenia who carried a CNV for schizophrenia, as much as I don't like that term, had poorer cognition than non-carriers. And this was particularly pronounced when the CNVs hit genes, so we say are intolerant to loss of function mutations. So that means are probably quite evolutionarily important. Another area in which CNVs are important is physical health. And it's these results which are perhaps more readily translatable to the clinic. Now, I can't give you an extensive list of all the physical phenotypes of the 53 CNVs that I've studied so far, because we'd be here until Christmas. But I can tell you a little bit about the kind of thing we're talking about, and also about one particularly important CNV and particularly well-studied CNV, which is the 22q11 deletion. This deletion is found in from about 0.5% to 1% of people with schizophrenia. So that's not an inconsiderable portion of our patients. And there are an awful lot of people walking around with this deletion who don't know that they have it. And it's the exemplar of the multisystem CNV. Physical phenotypes presented in childhood are these, so things like immunodeficiency, congenital cardiac malformations, hypocalcemia, palatal abnormalities. But it's not that infrequent that some of these get missed. So I've seen adult patients whose immunodeficiency, which must have been present since they were a child, has gone undetected until they're an adult and they have schizophrenia. Similarly with hypocalcemia, this can cause particular problems around hospital operations because it appears that their calcium plummets even further, and it can cause major problems. And in adults, this CNV is associated with an early onset of Parkinson's disease. Now, aside from the kind of syndromic congenital anomaly kind of stuff, There's emerging evidence that this CNV also plays a role in what we would consider to be your more everyday problems, such as type 2 diabetes. So some recent work which has come out of the Toronto clinic has found an increased risk of type 2 diabetes with an odds ratio of about 2.5. But interestingly, with an age of onset of 18 years younger than people who don't have this CNV. Now, going back to saying that 0.5 to 1% of people with schizophrenia have this deletion, that has a considerable impact on how we should possibly be treating these individuals. Now, as I mentioned the Toronto clinic, so it's a clinic run by Professor Anne Bassett, and they basically have been working on 22q for decades and have got a huge population of these individuals who come in regularly for follow-up, and so they have longitudinal data. And they've developed some practical guidelines for clinicians to suggest what you might be able to do to try and maximise the care of people with this deletion. There are paediatric and adult versions, and I'd very much suggest looking at least at the adult ones, because it really provides some quite neat guidelines as for how often you should be doing certain blood tests, should you be doing an echo, things like that. So it's potentially quite relevant in general adult psychiatry. So forgive this chart, it's quite hectic, but I'll unpack it a little bit in a second. Studies have also been carried out examining neurodevelopmental CNVs and links with medical conditions, the standard medical conditions that we would see fairly regularly in people without a syndromic presentation. So Karen Crawford et al. from Cardiff took the neurodevelopmental CNVs that I'd looked at previously and included the reciprocal deletion or duplication. So for instance, 22q deletion, she added in the duplication at the same time. So you're looking at a loci-wide association. And she looked at an association with 58 common medical phenotypes in the UK Biobank. Now, I said obviously that this is a slightly hectic chart, but basically it's ordered by significance level. So your associations at the top are have the strongest significance. And actually my screen's hiding it, but I think hypertension is the one at the top. But there are some others, such as obesity and COPD, which keep coming up again and again for these CNV loci. And by and large, these people will not know that they have these CNVs. Okay, so I've spent some time outlining CNV associations with psychiatric disorders and related phenotypes, including physical health. But something that I'm often picked up on when I do this kind of talk is, yes, but how are you going to translate it? And the truth is, we already are. So I wanted to tell you a little bit about what we're trying to do. During, I think it must have been towards the first lockdown, which was very helpful, we started our first psychiatric genomics clinic in Cardiff. And the clinic is staffed by Professor James Walters, who's a consultant psychiatrist and an academic, Dr. Annie Proctor, who's a consultant in clinical genetics. Donna Duffin is our genetic counsellor who's recently started with us. I work in the clinic as part of my special interest time, and we have an MDT once a month, which is staffed by psychiatrists from all of the different bits of psychiatry, including intellectual disability psychiatry, geneticists, sociologists, ethicists. You know, it's a really big. It's quite a nice MDT, and that's where we bring cases and discuss how we're going to approach individuals. So we provide input for individuals and families, basically who have any questions about potential genetic risks to them and their families if they have a family history of mental health disorders. We talk to them about risks associated with known neuropsychiatric genetic variants, so the ones that we study, whether that's ascertained via the NHS or via our research. And we discuss with them the implications of a diagnosis of a neurodevelopmental disorder. Another question that often comes up, which is probably not an easy answer for just yet, is what should prompt you to think of CNV testing? The first thing is to say that you don't necessarily have to test everybody. There's more to genetics than testing. Genetic counselling is so important. And there's some evidence from, I think it's from Janine Austin's group in Vancouver, which suggests that just talking to people about the familial basis of their disorder and the genetic etiology gives them some benefit. You know, they understand more about the disorder. That being said, what about genetic testing? So neurodevelopmental arrays are widely available. I'm sure they're available in every NHS trust in the UK. And you should check your local referral criteria. But certainly in Cardiff, the kind of approach we are taking and what makes me think of CNV testing is if someone has more than one neurodevelopmental phenotype. So to give you an example, and I use schizophrenia because that's what I deal with mostly. If I have someone with schizophrenia and ASD or epilepsy, or perhaps some kind of congenital anomaly, or an extensive family history of these things, that's what makes me start thinking, hang on now, do we have a highly penetrative genetic variant behind this, which might give a partial explanation for etiology? I want to add one little bit about this because it's come up recently. Common variant genetic testing is not that useful right now, so it's not really clinically available or of an awful lot of utility. It will, I think it will actually become helpful in probably in the areas of treatment choice, establishing risks of side effects and things like that. There are some companies who are offering genetic testing for common variation. And I would suggest caution before diving into anything. So I've heard of a couple of these companies coming up recently. Try and see if you can get to the bottom of what research is underlying whatever test they're offering. And was that research done in the same population as your patient? So vast majority of the research in psychiatric genetics has been done in European populations. And we are trying to increase diversity and improve that situation. But if your patient is not from that population, then that really limits how much you can apply some of the results. And I think, yeah, a lot of people don't necessarily think of that when they see all these kind of nice publicity materials from the genetic testing companies. But when in doubt, ask, and ask around if anyone's got experience with these companies. Okay. Now, not an inconsiderable amount of the time, when I give this kind of talk, I get concerns raised about the ability of our patients to tolerate genetic testing discussions. And some people seem to worry that we will cause our patients distress or even considerable psychological morbidity just from the discussion. And so during my PhD, I spent quite a lot of time looking at the literature available on this. The literature on genetic testing in those with psychiatric disorders on their own is not extensive at all. But what has been done suggests that whether you have a psychiatric disorder or not, there isn't a considerable negative impact in terms of psychology from having this kind of test and from receiving a result. Others have raised concerns that those receiving genetic results might engage in fatalism or genetic essentialism. So in this context, fatalism might mean that someone who knows their CNV carrier status might experience a loss of control and believe that their CNV guarantees that they're going to be unwell forever. And so there's no point in taking their medications. However, research from psychiatric and non-psychiatric populations suggests that this just isn't true. So attributing etiology to genetic factors doesn't result in fatalism. And if anything, it seems to result in a greater endorsement of medication. So they seem more likely to take their medications. Genetic essentialism might mean that a CNV carrier would see genetic explanations as the be-all and end-all for their disorder, and this might increase their distress. but there's really little evidence that any of that actually happens. It's also worth pointing out that ethical concepts might actually push us in the direction of offering genetic testing. Individuals can only access potential tailored interventions, such as those related to physical health, if they know their CNV carrier status. And so that's the start of it for them. And of course, if we're going to continue to develop clinical guidelines, such as that from the Toronto clinic, the only way we're going to be able to do that is if people know their CNV carrier status. Otherwise, they won't be able to take part in research. Ultimately, I think the most important of these principles is autonomy, providing patients with the required information and basically allowing them to make their own decisions, of which the vast majority are capable. I wanted to end by sharing with you this study from the team at Geisinger Health, which is in Pennsylvania. They analysed exome sequencing data in conjunction with data from electronic health records, and it's a really nice example of a translational piece of work. So of their sample, 708 individuals carried a neuropsychiatric CNV. So they defined theirs slightly different to the way I did mine, but there's a large overlap. And out of those people, only 6% had a genetic diagnosis. So only 6% knew they had a CNV. 29% had a documented diagnosis of a neurodevelopmental or psychiatric disorder. And that was quite broadly defined. So it extended neurodevelopmental to include epilepsy, and psychiatric included things such as well as what you'd expect, also things like OCD and bipolar. They selected a number of CNVs for return to offer to patients. And this is something we've started doing in Cardiff, which I can tell you a bit more about if you're interested. They had 141 people who carried a returnable CNV. And they sent invitations to them saying, look, would you like genetic counselling to discuss this? And so we can tell you about the result. 86% responded, and then 60% of those went on to have in-person counselling, almost 20% had telephone counselling, and about a quarter had initial sessions, but they didn't really follow it up. And so one of the things we're doing in our clinic in Cardiff is one of our samples was consented for return of results. So I'm actually contacting people now, telling them that they may have a genetic variant and would they like a genetic counselling session to discuss whether they would like to pursue it further. And anecdotally from my work, it's all gone down fairly positively so far. But back to the Geisinger study, they carried out a qualitative study based on these people who had genetic counselling, and 95% of people had positive or neutral experiences. It was common for them to have already tried to explain their disorder away, and they often blamed their social circumstances or their life circumstances. And their participants expressed a relief and satisfaction at having some partial medical explanation for their struggles and for their disorders. Many of them expressed that their CNV result reassured them that the diagnosis wasn't their fault at all. And that is something that really rings true from my experiences. I have an awful lot of people who have that relieved sigh on finding out their results. Quite a lot of the people in this study said they wish they'd known sooner. And the vast majority plan to share the results with their family, particularly with the motivating factor being children and grandchildren. So, excuse me. To summarise, rare CNVs are associated with neurodevelopmental and psychiatric disorders to varying degrees, but they're also associated with related phenotypes, including some physical health things which have potential for translation into clinical care. Aside from all of this, patients are often interested in the genetic basis of their disorders and seem to derive a benefit from the discussion whether or not to test. There's very, very little evidence that engaging individuals, whether they have a psychiatric disorder or not, in these kind of discussions has any sort of negative consequences in the long term. And ultimately, I think this is an opportunity to empower our patients to make autonomous decisions. And so I see that as a positive thing. Thank you very much. And I'd be very happy to take questions.
Great. Thank you very much. That was a great talk. I really enjoyed it.